All right, well, hey everyone, I'm Sam Alcan, and welcome again to our COVID-19 and food safety office hours. Today, we're going to be talking about whole genome sequencing and how it helps us to identify and track outbreaks. Now, our focus today will be on foodborne outbreaks, but whole genome sequencing has also been critical for COVID-19 tracking and identifying the emergence and spread of variants. And on that note, we've seen the spread of a subvariant of Omicron, BA2, which has become the dominant strain in the US. Now, it doesn't appear to be too different from Omicron in severity of disease. And overall in the US, we are at the lowest number of cases we've been at in a year now. Whether that's an artifact of reduced testing is a question. And we are seeing instances where, relaxation of mitigation practices like lack of masks and distancing can lead to upticks in cases. Here at Cornell, we just saw that the other week when we removed some masking. And we're also seeing upticks, across Europe. So whether or not this is going to translate into another large wave, we'll see. My recommendation is always to be mindful of the COVID-19 cases in your area and adjust your mitigation practices accordingly. Now, in a minute, I'm going to pass this over to Dr. Martin Wiedmann, who's going to bring us up to speed on whole genome sequencing and its role in outbreak tracking. But first, I'd like to introduce our panel for today. We have Dr. Olga Padilla-Zakor, a professor of food science and the director of the Cornell Food Venture Center. We've got Dr. Martin Wiedmann, the Gellert Family Professor in Food Safety and the co-director of the New York State Integrated Food Safety Center of Excellence. We have Donna Clements joining us. She is a coordinator and the Northeast Regional Extension Associate for the Produce Safety Alliance. We've got Dr. Aliosa Trimick, who is an extension associate in our Dairy Extension Program and our COVID-19 resource guru. Today, we also have Dr. Renato Orsi joining us. He's a senior research associate in the Department of Food Science and does a lot of work on whole genome sequencing, data analysis in relationship to foodborne outbreaks. Great to have you. We also have Dr. Callie Neal joining us. She's a professor of microbial food safety in the Department of Animal and Food Sciences at the University of Delaware, where she does a lot of research into the transmission of public health and food safety relevant viruses and pathogenic bacteria. And I'm Sam Alcan, an assistant professor here in the Department of Food Science, the focus on dairy food safety. We also have representatives from New York State Ag and Markets, from both the food safety and dairy divisions on the line if there's any particular questions there. And remember, you can always ask questions. All you need to do is, if you're logged in, you can hit that little chat box at the bottom, type in your question. You can either send it directly to me or to everyone, however you want to do it, and then we will read it out. I know many of you have pre-submitted questions, so I think it's going to be a really great discussion today. So actually, I'll give Al a quick second to mention, are there any updates to the website? And then if not, we'll toss it over to Martin.
Yes. Thank you, Sam. Yeah, we have 5 small updates. So under food industry resources, we put a CDC COVID-19 isolation and quarantine calculator. It's basically a decision tree tool where you can go through a couple of questions and then do you have to isolate? Do you have to quarantine? How long? Is there anything else you have to do? Under tracking COVID-19, at the bottom you can find information on COVID-19 variants, and we put WHO statement on the new Omicron lineage BA2. And then under the same page, you can find peer-reviewed papers, and we put 3 new papers there. One is on how long does the person infected with the Omicron variant shed the virus, how transmission of Omicron variant is happening and how it's evading the immune system. And then the third one is on the effectiveness of Pfizer vaccine in people 5 to 17 years old. This is basically it when it comes to updates. If there's any questions or comments that anyone has, please use the contact list on the page. And that would be it from my side. Thank you very much, Sam.
Well, thanks, Al, for keeping an eye on COVID for us. So hand it over to Martin. Take us away. Bring us up to speed on whole genome sequencing.
All right, everyone can see my full screen.
Looks great.
Sounds good. All right, so I'm going to jump right in. So I'm going to talk about for maybe 25 minutes or so to leave ample time for questions about whole genome sequencing and outbreak detection, what we've seen and what we expect to see. I'm going to give a little bit of background first, then talk about the pathogen detection database, which is an important tool in this context, and then weave in some more case studies on that. I'm going to keep the background pretty brief. I will provide resources if someone really wants to dig in how it's done. There are plenty other resources out there, so I'm not going to regurgitate that information. Obviously, we're here because WGS use is, I'm going to call it rampant, but it's widely used now by regulatory and public health agencies. Sharing of WGS data is what I'm going to focus on, really has upped tremendously. It's so easy for people to see, get WGS data and interpret them. And sometimes that facilitates what I'm going to call WGS innuendo, where it's like, oh, this outbreak is caused by blah, blah, blah. And without really the full information there. And I want to sort of, that is a risk for food companies, and it's something people should be aware of it. And this data sharing also allows everyone to essentially armchair quarterback CDC and everyone else in these outbreak investigations, which I think is going to really make things interesting. The other reason why we're here and what I'm going to try to emphasize today is while whole genome sequencing is a really important tool, what we really realize is that the basic epidemiology is so important. Whole genome sequencing can and should not replace epidemiology. But I think often this is where actually industry is even less clear than with regard to whole genome sequencing, how it's used. So I'm going to give some examples of that and the importance of epidemiology. Whole genome sequencing for foodborne pathogen has really been used since 2013. We started with Listeria monocytogenes back then, and of course now we're sequencing essentially both human clinical isolates as well as food isolates and isolates from food environments for all key pathogens. The impacts of it are well documented, probably best illustrated by Listeria monocytogenes, where we went from detecting one outbreak every 3 years before we had any subtyping to 7 to 8 outbreaks a year with whole genome sequencing. 25 to 30 times more outbreaks detected. Not only did we detect more outbreaks, but we're detecting smaller outbreaks. Before subtyping, average outbreak about 70 people. Now we're talking about an average outbreak of about 3 to 4 people. What does that mean? Small companies are going to be more likely to be detected as a source of an outbreak. Even if you have very sporadic contamination, let's say you have a resident strain in your facility, but you only contaminate a low proportion of your product, let's say less than 1%, or only contaminated every few months. This could still be and will be detected as an outbreak, often an outbreak that goes over extended time, which really has changed the picture in terms of the number and the types of outbreaks detected, and the types of foods, and the number and types of companies linked to foodborne disease outbreaks in the US. And I think it's one of the challenges we have as we move into this new world. Sort of a brief level set on whole genome sequencing. Again, not detailed. We provide resources where you can dig deeper into that. The idea is that most foodborne pathogen genomes we deal with have anywhere between 2 to 10 million ACTs and Gs. And when we compare these, and here is what this would look like, we can find differences, small differences, like isolate 1 and 2 differs by 1 nucleotide, and that's what we would call 1 SNP. Single nucleotide polymorphism. If you look at 3 versus 4, they differ by 3 SNPs, the T here, a T here, and an A here. That's essentially, in its simplest one, how whole genome sequencing works. Of course, it's more difficult because there are a number of different approaches how to analyze data. We can do k-mer-based SNPs. We can do high-quality SNPs. We can do multi-locus sequence typing, which comes in 2 flavors, whole genome MLST and core genome MLST. When we talk about high-quality SNP, we talk about SNP differences. When you look at data from MLST, we talk about allelic differences. The numbers can be interpreted slightly different, but to be perfectly honest and keep it simple, the differences aren't that big, okay, in terms of our numbers. If 2 isolates on high-quality SNPs differ by about 7 SNPs, you would expect something similar by whole genome MLST. I'm talking order of magnitude. I'm sure some of the experts here, if there are some, are going to cringe, but for all practical intents and purposes, they're fairly comparable in the number of differences they give you. interpreting SNP differences is, where I'm sure we'll get some questions and then where things get really challenging. And people want simple answers. They want same or different. And there is no cutoff. I'm going to just point everyone, no one will give you a cutoff, and I won't give you one today either, which says, okay, if 2 bacteria are less than, insert random number such as 7, then they're the same. And if they're more than 7, they're different. Why is this? Bacteria can spread quickly. I can have a bacterium in my facility. It can get on someone's shoes, that goes in an airplane, flies 1,000 miles, and that same organism now is found in 2 locations. On the other hand, I can have locations where bacteria can grow very quickly. Let's take a chicken flock that's actively infected with salmonella, where these salmonella might replicate every half hour to a tune of 50 times a day. That means if I'm in that environment, these salmonella are going to become different very quickly. While if I have the same salmonella in a dry powder facility, where it might not replicate for days, it might take decades to accumulate the same number of SNP differences as you get in a week in a poultry house. That is one of the key reasons why we will never have a cutoff. SNP difference numbers are a starting point, nothing else, no more, no less, okay? And I'm gonna get into that a little bit more. There are numbers floating around. How did we come up with them? Here's one example. We mapped a lot of isolates, including outbreak isolates and non-outbreak isolates, and come up with this number of 7 here, okay? The left side of 7, less than 7, is mostly isolates from the same outbreak. If we go on the right side of 7, rarely do we find 2 isolates from the same outbreak that have more than 7 SNP differences, okay? There are rules of thumbs out there, and if you look carefully, that's what they're called. These rules of thumbs are just that. There are no real cutoff. The interpretation depends on what decision you're trying to make, what organism you're looking at, what metrics you're looking at, epidemiological data, and as I said, a big one is how fast do we think these organisms replicate in the environment that we're in? And the problem is we don't know it. We have an outbreak of 50 patients of salmonella. We don't know whether it caused it by chicken, at least a priori, or whether it was caused by a processing facility. However, that fact is shown and illustrated here, where the rule of thumb values put out in one presentation by the CDC actually differ for suspected foodborne versus suspected zoonotic coming directly from animals. And as you might have figured out from my words is, if in a zoonotic one, the cutoff is higher as a starting point, the rule of thumb. Why? Because the organisms replicate faster, therefore accumulate differences more rapidly. But then, here's another set of recommendations or rules of thumb from FDA, where it said SNP distances that support close relationships. They call it match. I would not use that word, but hey, they're neutral or they do not support. But the important point here is using SNP thresholds, and it applies to WG-MLST and core GMLST as well, alone to assess whether bacteria originate from the same source can be misleading. I would have been even stronger, is often misleading. And we're going to dig into that and show how. Okay, so there are rules of thumbs, but no one ever will give you a line in the sand where it's like same, different, because you can't. All right, so let's talk about the data sharing part of this aspect of where we are and how things have changed with regard to using these tools in food safety and for outbreak detection and beyond. The database we're talking about here is the pathogen detection database. You just simply type pathogen detection, NCBI, in your favorite browser, and you should be getting to this page. You click around a little bit and you find the number of genomes that are found in there for the different organisms. And what we can see is this database is, I would call it massive. Salmonella, more than 400,000 whole genome sequence data in the database. Now, this is everything. This is human isolates sequenced by CDC and others throughout the world. These are food isolates sequenced by FDA, FSIS, state health departments, and isolates sequenced by academic labs and sometimes maybe even commercial labs, okay? But these data are in there with varying levels of details attached to each whole genome sequence. E. coli, more than 200,000. Listeria, more than 50,000. Chronobacter, obviously in the news recently with the Chronobacter outbreak, clocks in at a measly 931 isolates. That's enough, to just scroll through them and take a look at those. And these 931 isolates are, grouped into 138 clusters. So clusters are groups of closely related isolates, way above those cutoffs we discussed. So this is typically, less than 50 or within 50 SNPs, the type of methodology used for grouping changes, but these are larger groups than the SNP cutoffs we talked about. So that's what's out there for everyone to click on and take a look at. Now let's take a look at it. So we're going to look at E. coli and Shigella, and I'm going to show you what people can find in there. And what we're going to start out with is just sort of showing how you can go backwards. And you can see this going backwards or going forward. So let's take from a while ago, from December of a couple of years ago, 2020, an E. coli infection that was sort of described like this. Lab testing identified an outbreak strain in a sample of Tenimura and antler romaine lettuce. And here's the interesting one, and this is why I picked this example. But investigators were not able to determine if people got sick from eating the contaminated romaine lettuce. Okay. So taken by itself, some people might be like, that's weird. I mean, we found a strain in this lettuce. We found it in people. How can we say we don't know whether people got sick from eating that lettuce? So let's first take a look at what that looks like when we pull up the phylogeny, the phylogenetic tree on this database. So we can see what looks like the clinical isolates here, 4 environmental isolates here from Michigan, romaine lettuce. So usually this is how you can cross-reference that. If you know which state isolated the isolates, what it's called, you can, through a number of search options, find the tree that represents these isolates, okay? And again, we'll share sort of a tutorial how to do that if someone wants to do this yourself. So very closely related, less than a couple of SNPs difference probably, that's what it looks like here. But we fail to claim, or no one wants to claim, that this company is responsible for it. Why would that be? We don't have epidemiology supporting it. Probably very few, if any, of these people said they ate romaine lettuce from this company. And even with, it's close to picture perfect as it gets WGS data, we're not always going to implicate a company as the source. Biologically, what could have happened? Well, this strain was found in a romaine lettuce. It got there from somewhere. Let's say water, solely for argument's sake. That water also could have contaminated something else. Someone else's produce, some other produce, some completely different products. Some cows could have drank that water, could have been slaughtered, and that E. coli was in ground beef. Many different ways. So may look surprising to some of us. I always think, well, as soon as there's a match, you're responsible for the outbreak. Even CDC and others, as illustrated in this example, don't always jump to that conclusion. Here's another example. This is also what we'll often see. Outbreak says E. coli infections unknown. Multi-strain outbreak is different from the other outbreak. Linked to leafy greens, unknown source 3. And there we just have the number of people hospitalized. And it says not enough information to identify the source of the outbreak. What does this one look like? Find a bunch of clinical isolates that all look closely related. And if we count up the ones in red that most likely fall within that time frame, it does add up to about that number that is mentioned here. So not all, lesson here, not all outbreaks that are detected as such, where we have multiple people linked together by very close related whole genome sequence data, is linked to a source, even with these sophisticated tools. So This obviously drives us to the conclusion, which many of us is pretty clear, but important nevertheless, is that whole genome sequencing alone cannot identify a source. If we have a cluster, a group of closely related isolates from different humans, it doesn't necessarily mean, with all the whole genome sequence data in the world, it doesn't necessarily mean we can say where it came from. Even if we find the same isolate in a food product as the first outbreak sort of illustrated. So reliable identification of the likely sources requires epidemiological and traceback data. Very often when I talk to industry about whole genome sequencing, there's a lot of question about SNP differences. How reliable are the methods? Questions like this. To me, it's almost the wrong question. The more important question is, how is the epi done? How are the traceback studies done? How do we know the epi is reliable to this? Has the epidemiology adjusted for these massive whole genome sequence data and working in that environment that we are right now? And when we look at this, we need to think about and look back, how do we find these elusive epi data to find likely outbreak sources? Classically, this was done with case control studies. Sometimes it was done with case studies, where case control is where we have cases and ask them questions what they ate, and then we ask controls, people that did not get sick, what did they eat, and find something that's common to the cases, but less likely to occur in the controls. We can use statistics for that. I still think of that as the gold standard. Case studies, essentially I'm going to call it a labor and money saving approach, where we compare one case to another case, but these are cases that are not linked to the same outbreak. So I don't need to go out and interview a bunch of controls. I can use the data that I already have for this. When even this is now possible, and today that seems to occur more often than not, we look at historical food consumption data. We can say, well, if we ask 30 people what they ate over the last 1 week, usually we have about 50% saying lettuce. If we have a cluster where 90% say they ate lettuce, it's probably lettuce. And maybe not quite as cavalier as I described it here, but almost. We all know, particularly those in the food industry, that this can be misleading. It can be misleading because consumption patterns change, sometimes very rapidly, and they probably changed even more rapidly with COVID. This can be particularly challenging when we implicate specific companies, because we might say, so 90% ate lettuce and half of them mentioned company A. So it must be company A. Well, what if company A has a market share of 70%? Half of people that say lettuce mentioning company A with a large market share may not be meaningful. It might point us in the wrong direction. To me, these are the questions food industry needs to ask. It needs to work with CDC and others in order to improve the epi data collections to be more reliable and help us to better identify likely food sources and do traceback. Now, this may sound like there are a lot of shortcomings to whole genome sequencing. Whole genome sequencing and the pathogen detection database are still extremely useful. And there are some things industry can and should do now. Okay? And one of them is if you have any isolate from the past linked to your company, to one of your facilities. And that can be directly, FDA found an isolate 10 years ago, or indirectly, that maybe an isolate was found close to in a harvest area where you harvest a lot of your produce if you're in the produce industry. Set up a watch for this isolate. You cannot set up in this database a watch where you watch isolates and get emails when new closely related isolates are added. Again, we have some more detailed instructional videos for that I'll sort of share at the end. But briefly, here's what you do. You have to have a login. You can use your private email address or anyone that you feel is not tracked. You identify the isolate you're interested in. Let's say, hey, there were a bunch of outbreaks and issues in Arizona, which often were one specific sort of type of E. coli. You find one, like I did here, environmental 2018. June 2018, I mark it in red. I simply click watch. That means, and then I can name it and I can say Arizona water isolate 2018, whatever I want to. I will select what I'm watching for. So here I said I watch for any isolates that's been in 30 SNPs of this isolate. And I save and off I go after I gave it a name. And then the day later or a year later, I will get an email like this, new E. coli isolate notification for watched isolate. Send me what isolate it is, Yuma, Arizona water, and it tells me that a new isolate or new isolates were added. In this case, 2 more clinical isolates were added. I, at any time, probably have like 15 or 20 watches set up for different isolates that are of interest to me for some reason or another. Okay, very, very powerful tool, which can help you very quickly get an early warning if something happening. I'm going to be honest, in some cases, I, through this mechanism, can find a new isolate being added to a cluster before a company will hear it from FDA even, because these data get deposited so quickly into this database. And then you can, from this email, click on it and you can see where exactly the new isolate was added in that tree. It's conveniently highlighted in red for you. So it's a very slick way to track isolates, okay? So now let's move to some of the recent outbreaks and other examples, because I think they're really illustrative. I think what we might see more, and I think it will hopefully get all of us to think a little bit about what the future holds. And not to pick on produce, but those were 2 of the interesting ones, and Listeria is close to my heart, the 2 Listeria outbreaks. I'm going to talk about 2 Listeria outbreaks. The one that's been proclaimed as linked to packaged salad produced by Dole. Again, you have the data that you can pull publicly. This is all public information from the food safety alert. And it talks about Listeria on equipment used to harvest iceberg lettuce. FDA conducted whole genome sequence and found the Listeria on the equipment. So when we used all this information, we found this cluster here, where we have 2 isolates one called garden salad, the other one from lettuce, one Georgia Department of Ag, the other one Michigan Department of Health and Human Services, which in this release on this webpage are listed as 2 of the states that found Listeria in this product. So that tells us we're probably right that this is the correct cluster. We find a number of clinical isolates here, which matches up with the number of clinical isolates tested. We find a large number of environmental swabs and bagged salad mixture that, unless you have detailed sort of internal information, it's hard to derive where exactly they came from. Yeah, this is about the level of information you get. US, Arizona, FDA, environmental swabs. So that really doesn't tell you that much. Another thing I want to point out while we're here is that for clinical isolate, all you get is US clinical. It doesn't tell you which state, and that is to protect information, potentially confidential information, and not allow people to cross-reference it, which is a weakness for industry. I think industry might only distribute into certain states. The date here is really the date when the isolate was added, the information was added, is not when the case occurred. For the US, for this human clinical data in the US, at least currently, you typically don't even get a year. When the case occurred, now we typically can deduce that the sequencing happens pretty quickly. So if an isolate was added in, on December 5th, 2019, that person, the sample was probably taken from that person sometimes anywhere from 2 to 4 weeks before then, best guess. And in the Q&A session, if someone has better information than that, feel free to correct me. So that's what you get, and what everyone can get. No privileged information, everyone can get this. Now, where this outbreak and the other one I'm going to mention what's going to be interesting is that when you look on top of this, something I cut off in the other slide, you see some more isolates, and they're called environmental other US, California, FDA water sediment. And some of them were added in 2021, that's when they were sequenced, but were isolated in 2012. But they're relatively close related, which is sort of like, oh, that's interesting. So I do what everyone did. I click on one of those and I get to this page here. And this is essentially the detailed record for any of these water isolates. And what we find here is a hyperlink to latitude and longitude. You click on that, and it gets you to Google Maps. And you very quickly, literally within 3 mouse clicks, can say, wow, there are these isolates closely related to this outbreak linked to Dole that were obtained from this creek, Alisal Creek, that flows through Salinas, and it's pretty close to the In-N-Out Burger and right off East Market Street. Now, all of us who are familiar with Google, we know it doesn't stop there. If you look at another one of those isolates and take it from there and just click on satellite image, you can see, wow, look at this isolate. It was isolated off the bridge over the slough here. And I can actually visualize what to me looks like some produce fields here. And it's, Salinas, outskirts of Salinas. So what else is going to be grown there other than produce? So I can map this to a tremendous level of detail with the data available there, which were sequenced by FDA. Does not look like to me these were isolates collected by FDA, but that's the type of information that's available in the public domain, but also, of course, to CDC, FDA, FSIS, state health departments, and everyone else. And I'm not sure everyone in the food industry realizes the amount of information that can be linked together with these databases today. Just briefly, second outbreak claimed to be linked to Fresh Express, okay? We can do the same thing. Michigan Department of Ag identified the outbreak strain. Look for it, here it is. And here it gets even more interesting because we have these environmental isolates from water sediment, in this case, collected in 2016. This was sediment collected over multiple years in a research project linked closely to clinical isolates, some of them with one SNP difference. Environmental isolates, water sediment, 2016. Clinical cases that occurred anywhere from 2016. So here we actually have some date information to clinical isolates that appear to be contemporary 2020, 2021. So 5 years after we found this Listeria monocytogenes in water sediment, it shows up in a bunch of clinical cases. And actually less related to it, as shown by this longer branch here, also found in these Michigan Department of Health and Human Service lettuce isolates. Which of course then makes some of us say, well, how come all these outbreaks seem to be close related to water sediment isolates? Well, lo and behold, there are more than 1,100 whole genome sequence data for water sediment data in this database. So it doesn't take someone, I don't know how to phrase it, believing into conspiracy, people with a crystal ball to say, well, if more and more data like this get deposited with WGS and GPS coordinates, how easy will it be for in the future for people to say, oh, we find a match of these human cases to an isolate in the environment with these coordinates. Let's look at anything close by that could have led to an exposure, whether it's a food processing facility, an area where raw material is produced, and so on. And this is what I see as part of the future that's coming at us pretty rapidly and maybe a little bit under the radar for some of us. And that's one of the things I wanted to point out. Now, application of whole genome sequencing doesn't stop there. The debates about SNP differences don't stop there. And this is, again, an older example, but I think it illustrates it. It's from an FDA 483. I should have cited that, but I didn't. Talking about a facility that was resampled. And here's sort of in red, I highlighted the key part. Whole genome sequencing matched some of the Listeria findings genetically to samples collected during the June-July 2016 inspection, as well as to samples collected during the 2015 inspection. So clearly, this facility has been visited multiple times by FDA, 2015, June, July 2016, August 2016. And every time they come, it sounds like they find the same strains of Listeria in this facility over a total of a 2-year period. Here it seems that it's 3 different strains that have been consistently present in this facility. Now, it doesn't suggest anywhere here that Listeria was found in finished product necessarily, but it does suggest that there's some evidence that this Listeria may persist in this facility, which in some points could be interpreted and accepted. This facility is really not operating under sanitary conditions, which is a short step to calling it insanitary conditions. And all of us in the food industry know that food that is produced under insanitary condition is adulterated. And you cannot put adulterated food into commerce. So this is how whole genome sequencing can lead to a recall of product produced in a facility like this, even if there may be no evidence, no data, no positive tests on finished product. So whole genome sequencing at the same time as we have all these publicly available data where we can even find information like this really, also is in my mind, and that's a conversation with some people, I'm not the only one thing, really redefining how we define adulterated foods, which obviously is something that food industry is and should be paying close attention to. So brief take-home messages, whole genome sequencing is a game changer. Not only do we detect more and smaller outbreaks, we better identify microbial persistence, and we can better characterize pathogens. I haven't talked about it, but it's an important use of these tools in the food industry. You have a Bacillus cereus, or what you think is a Bacillus cereus, is it really? The number of WGS data sets, including from non-food and non-human sources, these sediment samples as an example, are increasingly available in the same database. And how they're being used and how they're going to be used in the future, it's really not completely clear. I think they're going to be used, they're going to be part of the decision making, but how, not quite clear, but it's something I think we need to pay close attention to. But the key take-home message here is whole genome sequencing are not magic bullets. They're not going to implicate a company as a source of an outbreak, even when you have really close matches, because we need good epidemiology and we need to understand potentially complex contemporary supply chains. What can and should industry do with this information? Sort of a few ideas I had. Make sure someone actually knows how this database works. If it's not someone in your company, have someone that works with you on it, whether it's someone at your university that you're familiar with, or someone or a consultant, whoever it may be. But to ignore that these data is out there would be dangerous, I think. Set up alerts for WGS clusters of concern. Many of you are like, well, we don't have anything that we're concerned about. I would rethink that, because there's so many data out there. You want to have at least an understanding what might be out there that should be relevant to you, whether it's from your facility, from the area your facility is in, from your raw material, or from the area where you get your raw materials from. Have a plan, or better yet, standard operating procedures. What do you do when you get a call from FDA and CDC? Important public service message. It is okay when CDC says, hey, we want to talk to you tomorrow because we have some cases that might be linked to your facility. It is okay. It's highly recommended that you bring in an outside consultant to help you with this. If you don't know how to interpret WGS data, if you don't know how to interpret epidemiology data and want someone to help you, call someone. Think about who that person is beforehand, okay? We have played these roles. We can help you find someone. There are people out there that can help you with this. And then plan and decide whether you may use WGS or whether you may not use WGS, but whether you feel like, well, maybe we do need to do some subtyping. So we identify if we have the same Listeria in our facility over time before someone else, before FDA, FSIS, or someone else finds it out, and before we have to defend that we are, whether or not we're operating in insanitary conditions. Here's a list of additional resources, some more in-depth presentations I gave, webinars that really focus on how to do the searching, how to utilize the database, and then some real background on the different methodologies. What's the difference between core genome and whole genome MLST and stuff like that. With that, Sam, I turn it over to you to moderate the questions.
Great, thanks, Martin. It was a great presentation, really informative.